Today, we're diving into the fascinating rulebook of life itself, genetics. We're going to explore how traits get passed down from one generation to the next, and more importantly, when those rules get broken. I mean, you've probably heard someone say it, "You have your mother's eyes," or "That's your father's smile right there." Well, guess what? It's not a coincidence. It's all part of this incredible game called heredity, and the rules are written right into our DNA. So, where does this whole story begin? Well, believe it or not, it starts in a quiet garden with a monk and a whole lot of pea plants. This was Gregor Mendel, the man who first started to crack the code of heredity. Back in the 19th century, most people thought that traits from parents just blended together, kind of like mixing paint. But Mendel, through his incredibly careful experiments, proved that it's not that simple at all. He laid down the fundamental rules that, honestly, form the bedrock of genetics even today. Okay, so let's jump into Mendel's first major discovery, the rule of dominance. You can think of it as a power dynamic between genes, explaining how some traits can completely overpower others. Here's how it works. For every trait, you get two versions of a gene, one from each parent. We call these versions alleles. Now, if you get two different alleles, one of them, the dominant one, is going to take the spotlight and mask the effect of the other, which we call recessive. That recessive trait only gets its chance to shine if the dominant one isn't there to steal the show. Let's look at a classic example from Mendel's own work. He took a purebred tall pea plant. Its genetic makeup, or genotype, is TT, and crossed it with a purebred dwarf plant, TT. So what happened in that first generation, the F1? Every single one of the offspring was tall. Why? Because that T allele for tallness is dominant over the T allele for dwarfness. But here's where it gets really interesting. When Mendel let that F1 generation self-pollinate, that hidden dwarf trait suddenly reappeared in the next generation, and it wasn't random. He found this perfect mathematical ratio. Visually, what we call the phenotypic ratio, you get 3 tall plants for every 1 dwarf plant, a 3-to-1 split. But on the genetic level, the genotypic ratio, it was 1 TT to 2 TT to 1 TT. This precise pattern was a massive clue for Mendel, and it led him straight to his next big idea. So let's move on to Mendel's next 2 laws, which really explain how genes are shuffled and dealt, almost like a deck of cards. We're talking about the law of segregation and the law of independent assortment. The law of segregation is pretty straightforward, really. It just means that when your body makes reproductive cells, sperm or eggs, your 2 alleles for any given trait split up. They segregate. This means a parent can only pass on one of their 2 alleles for each trait to their child. It's a 50-50 shot which one it'll be. And this is so important. As geneticist Dr. Elena Torres puts it, when parents pass on genes, it's not just matching traits that matters. It's the very segregation of alleles that shapes genetic possibility. In other words, this separation is the fundamental reason we have so much genetic diversity. It's what makes every individual unique. All right, now for Mendel's next big rule, the law of independent assortment. This one basically says that the genes for different traits are inherited independently of one another. Think of it this way. Inheriting the gene for pea color has absolutely no effect on inheriting the gene for pea shape. They're not a package deal. This is a perfect illustration of what we mean. It's called a dihybrid cross, which is just a fancy term for tracking 2 different traits at the same time. You can see when a plant with yellow, round seeds is crossed with one that has green, wrinkled seeds, the next generation doesn't just produce the original combinations. You get all sorts of new mixes. Yellow wrinkled and green round pop up too. And again, it's not random. These new combinations show up in a very specific classic ratio, 9 to 3 to 3 to 1. This number is the smoking gun, the definitive proof that the genes for seed color and seed shape are being inherited independently, just like Mendel's law predicted. But what if the story isn't that simple? What if the rules aren't always followed? This is where genetics gets a little mysterious. What happens if some genes don't go their separate ways? What if they stick together? So that leads us to a pretty critical question. Is independent assortment always the case? Do genes always act like they're on their own? The simple answer? Nope. As fundamental as Mendel's laws are, they're not quite universal. The story of genetics, it turns out, has a few plot twists. And that plot twist started to unravel in 1905. 2 scientists, William Bateson and Reginald Punnett, were studying sweet pea plants. They were looking at flower color and pollen shape, and they totally expected to see that classic 9:3:1 ratio. But they didn't. Their results were way off. Something else was going on. The solution to their puzzle was a phenomenon called genetic linkage. And it's pretty much what it sounds like. It's the tendency for genes that are located close to each other on the same chromosome to be inherited together. They basically break the rule of independent assortment. This diagram makes it really clear. Genes A, B, and C are all on the same red chromosome, so they're linked. They're much more likely to travel as a group. Meanwhile, gene D is on a different chromosome entirely, so genes A and D are unlinked, and they'll assort independently, just like Mendel said. But even linkage isn't an all-or-nothing deal. There's complete linkage, where genes are so close together that they almost always get inherited as a single unit. But then there's incomplete linkage, where the genes are a bit farther apart. This means there's a chance they can get separated during a process called crossing over. That's when chromosomes swap pieces of themselves, which can lead to some new combinations. So, we've seen Mendel's rules, and we've seen how they can be broken. The big question is, why does any of this matter to us today? Well, the answer stretches all the way from Mendel's quiet garden right into the heart of modern medicine. Understanding these rules and their exceptions is absolutely critical. It's how we breed better crops to feed the world. It's how we understand and predict genetic diseases. In fact, scientists use the concept of genetic linkage to create genetic maps, which help them pinpoint the exact location of disease-causing genes. All of this is fueling the development of personalized medicine and helps explain the incredible diversity of life on our planet. In the end, the language of our genes is really a story full of rules and just as many fascinating exceptions. It's the story of who we are. And the most exciting part is, we're still figuring it all out. It really makes you wonder, what will this story teach us next?